Okay,
Thank
let's
you very
start.
much.
A very good
Very
morning,
much.
everybody, and welcome you all to the 17th webinar of Plant Breeding Webinar Series.
Good
These
morning,
webinars are
ma'am.
being organized by the Foundation for Advanced Training in Plant Breeding on a monthly basis. And today we have Dr. Mohan K, who will talk about BT resistance in pink bollworm. The purpose of these webinars are to sensitize people to know and value the importance of plant breeding, and also to create an awareness on various aspects like technology integration, policy support, regulatory overview, farmers' comprehensions, and various other aspects required for inclusive development of agriculture. Foundation for Advanced Training in Plant Breeding, abbreviated as ATPBR, is a non-profit organization which aim to promote advanced technology in plant breeding and develop qualified plant breeders who would complement and add value to the existing system. Our efforts are toward creating a conducive ecosystem where skillful organizations or resourceful persons work together to develop joint courses, research, co-incubations. ATPBR organized professional trainings, workshops, and various awareness sessions to build required competencies of the global standard. And we also organize annual conference named as 5F Farming Conference in different locations across India. Now, I would like to take the privilege to introduce our moderator, Dr. K. Narayanan, who is the founder, director, Thaika Seeds, and director, CEO, Agri-Genomics Lab. He is also founder director at ATPBR. He co-founded Metahelix Life Science that was acquired by Rallis India Limited, a Tata Group company, in 2016. He later co-founded and chaired the board of Kottaram Agro Food, a food technology enterprise that make packaged food product under the brand name Soulful using traditional Indian grain like ragi. This company was also recently acquired by Tata Consumer Product Limited. Dr. Narayanan has been a member of several committees and advisory bodies, including several task force and working group of the DBT, Government of India. He is involved with several government initiatives that promote entrepreneurship and mentors several startup companies. I request Narayan sir to take the floor. Narayan sir, over to you.
Thank you, Abarna, for the very generous introduction. Good morning to all of you. I think my greetings also on the occasion of Azadi Ka Amrit Mahotsav. I think what we are going to discuss today is a very important topic. I think all of you will agree that over the last 2 decades, if you consider the agricultural sector, one of the biggest technological revolutions has been brought in by the BT cotton technology. While the technology was being developed and deployed, even from the initial years, there was a realization that there is potential for development of resistance among the target insect group, which is the bollworms. 3 of them are major, Helicoverpa, Aries, and the pink bollworm, about which much of today's discussions will happen, Pectinophora gossypiella. So there was a refugia strategy that was deployed or envisaged for deployment. Whether it was effectively deployed, I'm not too sure. Thankfully, I think the other 2 bollworms, Helicoverpa and Aries, have not yet shown the extent of resistance development as we have seen in the pink bollworm. And to talk about this, so this resistance is actually threatening the very sustainability and the value of the original BT cotton technologies that have been released so far in our country and elsewhere. And so we need to move on in terms of leveraging technology and science in trying to address this problem. I think the refuge in the bag has been thought about, and that could alleviate or arrest the further spread of the resistance, but resistance is already present in much of the population of pink bollworm in the country. And who is a better person to talk about the resistance and what we can do to arrest it or at least manage it in future than my old friend and old colleague, Dr. Mohan. I think he has been a very key person in the stewardship of the BT cotton technology in the country ever since its deployment, first deployment in 2002. Dr. Mohan had his degree and postgraduate degree from University of Punjab, if I remember correctly, and then he had a stint at the Indian Institute of Science, and I think his PhD work was on viruses and insects. Am I correct?
Yeah, absolutely.
Okay, and then he had I think a decent stint in the ICR system. He was with a scientist with CPCRI, Central Plantation Crop Research Institute, and later in Indian Institute of Horticulture Research. I'm not sure if some of the participants that we have here were his colleagues at IAHR, because I know some of them were at IAHR. And then he moved on to the Monsanto Research Center in Bangalore. And I think, like I mentioned, he was a key person in ensuring that the converted lines into BT is having the right levels of expression, monitoring the expression levels in the field, actual hands-on work, not a very glamorous work, but a very, very critical component, which many people don't hear about. I think he has also been in the forefront of tracking the resistance development in the various organisms. Pink bollworm resistance, when it really started showing its head in the Gujarat region, I think he was right there trying to collect, and many, many achievements to his credit in this area. I think one of the things that I remember was his ability to culture the pink bollworm in the lab, and this being almost like an obligate pest, even though some people say that there are some alternate hosts for the pink bollworm. Very difficult to culture it in a lab situation so that you can do some bioassays and quickly, like the other two bollworms, I think this one is a little recalcitrant. So he can be credited with leading the team, which put together a proper system for the culture of these larvae, sufficiently enough so that you can do some bioassays and get some preliminary information. So rather than taking more time myself, I think I would let Mohan take you through his presentation. We have many, many stalwarts in this area in the audience, like Dr. T.M. Manjunath, for example. And I would expect that we will have at least 10 to 15 minutes of discussion after about 40 to 50 minutes of Mohan's presentation, when we can leverage the experience and the opinions of the people in the audience. So my suggestion is, Mohan, you take about 40-45 minutes for your presentation. Will that be fine?
Absolutely. That should be fine.
Okay. And then we will have about 15 to 20 minutes of discussion. And just to bring some order into this discussion, being a virtual forum, if anybody has a question, they can either post it in the chat box or show up your hand. You can raise a hand. There's an icon in the screen which you can use to raise a hand. And in any case, I think the time is 11:10. We will try to close by 12:20 or 12:25. Okay, over
Sure.
you, Mohan.
Thank you. Thank you. Thanks a lot. And greetings to the 71 participants in this webinar, and greetings to ATPBR. And I would like to thank Dr. Aparna Tiwari for the formal invitation to be a part of this webinar series. And informal invitations, and it was Sharan Angadi who got me interested in the activities of ATPBR. So thanks a lot to all of them. And probably I will start projecting my slides. Can I?
Sure.
Please go ahead. I think it may be useful if everybody puts their microphones on mute. And just to make sure that we have enough bandwidth, you can probably switch off your videos as well. We can come back to that when we have the discussion session.
Can you see my slide?
Yes, yes,
Yes, sir, we
very
can.
well.
Okay,
Go ahead.
thank you. Thank you very much. Thank you very much. And thank you for all the generous and nice introduction, Dr. Narayanan, my colleague of good times. No, it's not that we are having bad times now, but yes, we really enjoyed our work together at Monsanto. Thank you very much.
Colleague of the best times.
Yeah, the best times. I totally agree. Okay, so I'm going to, today I have structured my talk to sort of discuss the current scenario of BT resistance in pink bollworm. Now, this BT resistance in pink bollworm and consequently the increased damage to BT cotton, which the farmers have been experiencing from 2014, when the first report of resistance to Bolgar 2 came in from one particular Bolgar 2 field in Amreli district of Gujarat. And since then, I think the BT resistance in pink bollworm has been a thorn in the flesh of the BT technology. It has really eroded the value of BT cotton technology. And let's see how we can kind of do a remedial action to set things right. See, currently BT resistance has been reported in pink bollworm populations across the 8 major cotton states. And this involves 12.5 million hectares, not a small number. And this has directly impacted the productivity through loss in yield and loss in quality of kapas, because pink bollworm infested bolls leave a lot of stains on the kapas, on the fiber, and this fetches a much low market value for the farmer. So the farmer loses on that front, and also loss in yield. And the cost of production for the farmer has gone up because he has to spend money for the control measures for pink bollworm. So it is a double whammy for the farmer, and he has been suffering from the last 2014. Though we take a lot of pride in saying that India is at the top spot in cotton production, but believe me, this is because of the sheer acreage. We have a large acreage of land under cotton, and the productivity in India is woeful, among the lowest in the cotton growing countries. And we are not really in a happy situation with respect to combating the pink bollworm menace. And this pink bollworm incidence has given a wake-up call to all the IPM measures which the cultivators have been ignoring all these years. And they have now, a concerted effort has been initiated in most of the cotton growing states to get the farmers to practice IPM measures. And also we have the recent, campaigns for, the mating disruption methodology. But again, all these things have a long way to go. These are powerful methodologies, but all these things have a long way to go because the homozygous resistant pink bollworm population is at an all-time high in all the fields. As a result, we are seeing early incidence of pink bollworm right at the bloom stage. At 45 to 50 days, when cotton starts blooming, pink bollworm appears. I mean, such is the large population transfer from the previous cotton season to the current cotton season. Now, what do we do about it? Unfortunately, at this juncture, we do not have a major impact-making technology in hand. I repeat, we do not have a major impact-making technology in hand, like the way BT cotton impacted, plant protection in the early days of its introduction. And nor do I see any technology in the near horizon, the near horizon of, say, 5 to 6 years. So what do we do with this? So with this backdrop, I kind of plan to map the technology landscape available for combating BT resistance and pink bollworm. And probably in the last few slides, focus sharply on the feasibility of developing a next generation BT cotton. In my opinion, I repeat, in my opinion, this is the only way out in the short run. Now, it's very heartening to see, news media reports like India to maintain top rank in world cotton production till 2030. That's something very heartening. But the truth is, like I said before, this comes from a large acreage. India has the largest acreage of cotton. But our productivity is really woeful. And so despite a rise in area, cotton yield drops to below 500 kilograms per hectare in India. Now I have encircled two figures, one of 566 kilograms per hectare. This was the maximum yield we got in 2013 and 14 during the peak, the peak season or the peak times when BT cotton was very effective. And now it has, in the last few years, it has slipped below 500. This is really, really worrisome. And the crop damage by BT resistance pink bollworm has added to the existing woes of productivity. Just to process check, am I audible?
Yes, Mohan, we can hear you clearly.
Thank you. Now, why do we need a fresh technological breakthrough in pest management of cotton? Why do we need it? Number one, even though we sing a lot of praise of the PT cotton technologies of 2002 and 2006, which has enabled this country a quantum leap in cotton production in India. But the BT cotton technology, especially the Bollguard 2, is about 16 years old. And pink bollworm has evolved resistance since 2014 and has spread throughout the cotton growing states, impacting the productivity and quality of kapas. But BT cotton is still good for the management of Helicoverpa armigera, but this may not be for long. If you remember, Helicoverpa armigera was a Khalnaik during the pyrethroid era of 1980 to 1990. This was a Khalnaik. This is because this organism was the monster during the pyrethroid era. It had gained resistance to pyrethroids and a host of other insecticides, and the farmers were helpless. Then came BT cotton technology as a big relief. So, with the track record, which we have seen with Helicoverpa armigera, we have to be extremely careful. And this Bollguard 2 technology is kind of old for this organism. Why do we say so? I have taken some figures from the resistance monitoring reports of CICR, the annual resistance monitoring reports. And as you can see, is my cursor visible? Movement of my cursor visible?
Mohan, we can see it. We can
Yes,
see
sir.
it.
Yes.
Thanks a lot. So the cursor, I'm focusing at the pre-launch, years of 1999, 2000. It was below 1, the LC50 of Helicoverpa armigera, and this has been steadily rising over the years. And the data here, 2011, there is a considerable increase in the mean LC50. And also the fiducial limits have increased considerably. For example, the fiducial limit of the 2011 data is around 3.5 ppm as against 0.2 or 0.3 ppm of the pre-launch. This shows that the Helicoverpa populations are growing in tolerance to Cribonacea. And I'm sure this is a similar picture with Cry2AB2 tolerance levels. So we cannot take it for granted the control of Helicoverpa armigera in the future. Next, as we develop a technology, the sap-sucking pests of cotton has been left behind. And this continues to plague cotton productivity and their management. And, for example, this has been a bad year for whiteflies in North India. And the farmers are really helpless, spraying twice a week for the control of whiteflies. And whiteflies also brings in the leaf curl virus disease. So all this can be bundled into a technology which we need to develop. Now, why didn't Helicoverpa armigera, why has it not developed resistance to BT? I mean, considering its track record of, resistance to pyrethroids. The reason is because there are a host of, crops which can support the multiplication of Helicoverpa armigera at the same time when cotton is grown in adjoining fields. See, for example, the average land holding for cotton is about 2 to 4 acres. And so you have the crops like sunflower, groundnut, maize, chickpea, pigeon pea, sorghum, soybeans growing in the adjoining fields, and they provide the natural refuge for Helicoverpa armigera. And this is one of the reasons why this monster has not developed resistance to BT proteins in BT cotton. Because, the homozygous resistant population from bollgard cotton has all the probability of mating with susceptible population flowing in from these other crops. So, unfortunately, we don't have this kind of panoramic scenario of natural refuge for pink bollworm. Now, continuing on Helicoverpa armigera, we did a satellite mapping in 2004 and 2008. And what we found was the central cotton fields, that is Gujarat, Maharashtra, Karnataka, and Tamil Nadu, they have plenty of natural refuge in the form of these alternate host crops. To the extent the natural refuge forms about 20 to 50% of the acreage, of the area. That is a large piece of natural refuge. So even if you don't, plant structured refuge for Helicoverpa, no problem. The natural refuge has provided its contribution. Whereas in North India, the natural refuge was very, very low in proportion compared to the BT cotton area. It was negligible. So in fact, if we have to watch out Helicoverpa, it has to be there in the North Indian cotton fields. Now let's come to the Khalnaik of this talk. That is the pink bollworm, or Pectinophora gossipella. This has turned out to be the black sheep in the bollworm family. Now, this is a functionally monophagous insect. What it means is cotton is the only major host on which it can eat and multiply. Though okra is also a host on which it can multiply, but compare the proportions of okra grown in this country compared to that of the 12.5 million hectares of cotton. It is a minuscule proportion. So okra and the other malvaceous weed, they form a very minuscule proportion of the natural refuge. As a result, this insect has been under tremendous selection pressure to develop resistance. Now, it is not that this insect, because it has been exposed to a large amount of BT protein everywhere it goes, it has developed resistance. It is because the natural mutants which develops, and this is at a constant rate for any insect, they are better selected on BT cotton plant. That is the only thing. For example, the Helicoverpa armigera might also undergo the same amount of mutations to be resistant to the BT proteins in BT cotton. But since the the susceptible population is flowing in from the natural refuge, the resistance gets diluted in Helicoverpa. Whereas that opportunity for a dilution of resistance was not there for pink bollworm because it had nowhere to go excepting BT cotton plants. And the second issue is the feeding habits of the larvae, it is highly protected. It enters a bowl, develops till the 4th instar or 5th instar and stays within the bowl, or develops an exit holes and drops into the soil, and it overwinters in the cracks in the soil. As a result, this insect receives, the larval stages receive very low exposure to insecticides. So insecticides are almost useless for the control of pink bollworm, excepting when the eggs have been laid, and it takes about 2 to 3 days for the eggs to hatch. And the young larva penetrates into the epicarp of the green boll within 1 hour of hatching, which means you can, if insecticide sprays are given which are ovicidal, yes, there is a chance of killing the eggs, but not otherwise. And this insect also has a capacity to withstand adverse weather conditions. It overwinters. The fourth instar exits this thing, falls into the cracks of the soil, and overwinters till favorable environmental conditions appear for it to emerge and start its life cycle again. And another issue which also helps this spread of BT resistance is the transportation through infested seed cotton. So seed cotton, which is produced in an area, for example, Maharashtra, Madhya Pradesh, it is also transported to other states for ginning or for oil extraction. And that is how the BT resistant population spreads to other states. Now, if you look at the pink bollworm infestation window, normally pink bollworm during the initial effective days of BT cotton, or during the pre-cotton, pre-BT era, normally pink bollworm is a late infester. It starts infesting cotton right from 90 to 100 days and continues to 130 or 140 days, or till the cotton is harvested. Now, this is a normal infestation window. And within this window, what happens is the farmer already takes two good crops of cotton till 90 or 100 days when the pink bollworm infestation starts. This is under normal conditions. But now what happens is, with the advent of BT resistant pink bollworm, the homozygous population is so large. The carryover from the previous season of cotton to the current season of cotton is so large. Now pink bollworm has started appearing in the bloom. It has started appearing at the 45th or 50th day of the growth of the BT cotton plant. As a result, it gets an early opportunity to multiply within the bolls of BT cotton, and more number of generations are spent. Instead of 3 or 4, it is now spending 7 to 8. The population increases there. And so any control measure we take has to be, we have to keep in mind of the large homozygous population present in the field today. Well, there are two schools of thoughts on the origin of pink bollworm. One school of thought says that it originated in the Indian subcontinent of Pakistan, Bangladesh, and India. The other school of thought says, yeah, it also originated somewhere in the Southeast Asian region. Whatever it is, this pink bollworm spread from this center of origin to Egypt first, because Egypt is another major cotton growing country. From Egypt to Sudan, and from Sudan it went to the Caribbean islands and made its appearance in the cotton fields of USA somewhere in 1917. And that is the way. And so in the US and Mexico, it is treated as an invasive pest. It has come from the Southeast Asia and is an invasive species. Now let's look at the key cotton bollworms in India, which were well managed by BT cotton technology. Helicoverpa armigera, pink bollworm, and Erias vitella were well managed by the single gene Bollgard. Okay, and when it was displaced by Bollgard 2 in 2008 onwards, all the 4 pests, including the tobacco caterpillar Spodoptera litera, was well managed by Bollgard 2, which expresses 2 proteins, Cry1AC and Cry2AB2, and the first 3 bollworms were extremely sensitive to both the BT proteins expressed. Now let's look at the risk of BT resistance evolution, which is a function of the refuge availability. Let me repeat. The risk of BT resistance evolution is a function of the refuge availability to the different cotton bollworms, and this is assorted. For example, as I said, the Helicoverpa armigera, or the cotton bollworm, is polyphagous in nature, and there is plenty of natural refuge especially in the peninsular India and the western part of the country. Pigeon pea, chickpea, sunflower, maize, sorghum, tomato, chilies. I mean, they constitute almost 40% of the land area in central and south Indian cotton belt, whereas North India has a very low proportion of this natural refuge, and thereby, hence, this insect is to be put on the watch out list for gaining resistance to Bt proteins in North India. Coming to Pecnophora gossypilla, this is a functionally monophagous pest and known natural refuge, and as predicted, this gained resistance first. I mean, it won the race in gaining resistance. The third is Aedes vitella. Aedes vitella is nowhere to be seen these days, even though, like pink bollworm, it is also almost a monophasic pest. It can multiply only on cotton and okra, and so this is again an important insect to be placed in the watch out list. And the last is Spodoptera litera, or the fourth one, and this is also polyphagous in nature, and so we have some amount of natural refuge to provide the susceptible population. Now, let's come to the launch area. Now, Bt cotton was launched in India in 2000 on a strong technological foothold. The reason being, the Bt cotton, Bt spray formulations have never been used. So that means all these bollworms were never exposed to Bt formulations or the Bt proteins. And cotton bollworms in India, relative to their counterparts in the US, were more sensitive. Really, they were more sensitive to the Bt toxins deployed in Bt cotton, both single and the stacked versions. Now, coming to the table, let's focus on Pecnophora gossypilla, the pink column, the last column. The pink bollworm was extremely sensitive to Cryphonacea, and to the same extent, extremely sensitive to Cry2AB2. So when we started, I mean, we were on a strong technologically footing with respect to pink bollworm, because this insect was very sensitive to both these proteins. And let's compare it to the LC50 values with heliothines in the US. Now, Helicoverpa zea is a much more bigger monster than Helicoverpa armigera in India. And look at their mean LC50 value. It is 25 ppm, as against 0.2 ppm of Helicoverpa armigera basal line susceptibility. So, I mean, there is absolutely no truth in saying that, the testing should have been done before Bt cotton was introduced into India. No, there is no truth in this. We have a strong technology background when Bt cotton was introduced into India. Let's look at the evolution of pest resistance in pink bollworm, which is the major subject of discussion today. Now, let me see. I was involved in tracking Bt resistance breakout for the Bollgard as well as the Bollgard 2, right from 2008, and I could see all these incidents play out like a movie before my eyes. Okay, let's look at the breakout of resistance to the Bollgard, the dual gene Bt product. Now, the possible precipitating factors for resistance evolution in 2003. And 2003, resistance evolution was noticed in one Bollgard 2 field in Amreli district in Gujarat. The arrow shows that. And what could have been the precipitating factors for resistance evolution? One was high selection pressure for resistance evolution, because there was very low abundance of natural and structured refuge for pink bollworm. And the resistant strain of pink bollworm had large areas of BT cotton for multiplication because it has adapted itself to multiply on BT cotton. So it had almost, let's say, wherever bollgard was grown, that turned out to be the host area for the resistant pink bollworm. So resistant population size quickly ramped up because all other bollworms were very effectively controlled by bollgard, prior to 2008, excepting pink bollworm. So pink bollworm it had a large area of host to multiply on. And also in some areas in Gujarat, and especially the Narmada belt, known as the Khandesh, the farmers, they extended the cotton cropping period, and they also planted medium to long duration cotton. And cotton, pickings were taken till almost say 200, 220 days. And this provided extra opportunity for the resistant pink bollworm to increase in population. So the resistant individuals really ramped up between 2008 onwards and till 2014. Now the end season IRM practices were ignored. I mean, this constitutes the breaking the pest cycle by destroying unopened bolls, summer plowing to kill the overwintering larvae and pupae. So all these were kind of not practiced very diligently. As a result, the carryover of resistant population from one season to the other was diligently taken up. And this provided a big door of entry for the resistant population. And also the movement of kapas, infested kapas, to the ginning mills and adjoining the neighborhood districts and neighborhood states helped in the spread of resistance pink bollworm. Now the first picture shows a few districts infested with resistant pink bollworm in 2013, and in 2019 it had spread to Gujarat, Madhya Pradesh, especially the Khandesh area where extended cotton cultivation was practiced, and to the important cotton growing areas of Maharashtra, Karnataka and Tamil Nadu. And in 2022, as we learn, in the last 2 years, the resistant population of pink bollworm has spread to the North Indian cotton fields of Rajasthan along the canal irrigation, and to Punjab and Haryana. So now we have the resistant pink bollworm strain almost across 12.5 million hectares. That is a large area. Now, let us look at the BT cotton events in India during BT resistance evolution. We had the cotton hybrids from various companies, and but in 2009, Gujarat, which predominantly grew bollgard, the resistance to Crivon was noticed, and in 2010 it ramped up to a few districts in Gujarat, and in 2010 and 11 it ramped up to spread to the adjoining districts of Maharashtra and Madhya Pradesh, and a similar route was followed by the bollgard 2 resistance, resistant strains of pink bollworm. Now, how did the resistance evolution to Crivon begin in 2008 and 2009? And we believe, for example, in 2000, the red star shows breakout of resistance to Crivon bollgard in a few districts in Amreli. That was the center point. And coincidentally, the same area provided us the resistant bollgard 2 resistant population in 2014. So we believe the triggering factors was the no structured refuge for BT cotton and extended cultivation of cotton beyond 140 days. This provided several more cycles of pink bollworm larvae to multiply. And also Gujarat, was a host for illegal BT cotton, the illegal Crivon BT cotton, which was grown right from, I think, 1999 and 2000, 2 to 3 years prior to the approval of bollgard in 2002. And probably these, illegal BT entrants had a questionable BT trait and questionable expression in their plants. And as I said, the end season IRM practices were ignored in breaking the pest cycle. Now, this is a Google map which shows the spread of Bolgard, resistant strains. In 2010, it had covered almost all the districts in Gujarat, and it had spilled over to the adjoining Khandesh area of Madhya Pradesh and Maharashtra. And in 2011, we could detect Bolgard resistant strains in three the states: Gujarat, Madhya Pradesh, and Maharashtra. And soon, the Bolgard 2 displaced Bolgard, and this helped to quell the resistance to a large extent. For example, from 2010 onwards till 2014, no incidence of pink bollworm was ever detected in all these three states. And we believe it was not a monogenic source of resistance, and resistance development had taken place in parallel in several districts and several Bolgard fields, because when we sampled the cotton bolls in 2009 season, we found that 69% of the bolls examined contained large pink bollworm larvae, as if there was no Cryonaceae present in those bolls. The CICR criteria was 2 bolls per 20 bolls, that is 10% infestation levels. Whereas what we found was almost 69% of the bolls examined, and several of these bolls, several of these fields had almost 100% infestation. So we believe, I mean, the pattern of spread and the pattern of infestation levels led us to believe that there were several mutants of cadarin, which we will speak later, which provided resistance to Cryonaceae. And when we subjected these populations to diagnostic dose, let me remind you that 90% mortality of BT susceptible pink bollworm could be obtained with 0.1 ppm of Cryonaceae in the diet. 0.1 ppm of Cryonaceae. Whereas these populations which we collected from the field could grow happily in the presence of 10 ppm of Cryonaceae. Such was the extent of resistance. And we believe it was multiple alleles of resistance. Only this can suddenly ramp up the resistance population across the 3 states within a gap of 2 to 3 years. Now let's look at the BT mode of action in Lepidopterans. Now it is largely believed in most of the Lepidopterans, with most of the insecticidal proteins of BT. There is a primary binding of the activated toxin to the cadarin. Cadarin is a cementing protein found in the epithelial cells. I mean, it is also displayed on the epithelial cells. It also forms the cementing protein between this epithelial cells in the gut of the Lepidopteran, susceptible Lepidopteran insect. And the mode of action in Lepidoptera and Coleoptera is nicely explained in this kind of schematic diagram. In the case of Lepidoptera, the activated Cryonaceae proteins, they bind primarily to Lepidoptera, they oligomerize, and then they bind to the secondary receptor in the form of APN, which is aminopeptidase, or it is the alkaline phosphatase, ALP. So process check. Am I audible?
Yes, Mohan, we can hear you.
Oh, great.
Someone else is also unmuted. Please mute all of you, please.
Okay. So what happened with the Cryonaceae resistant pink bollworm? we had extensive mutations in the cadherins in the resistant alleles of pink bollworm in India. And there were multiple alleles, which means there was a mix of minor resistance and major resistance alleles, and that played havoc, and that gave result to the rapid spread of resistance to bole guard within a span of 2 to 3 years in 3 major cotton growing states. Now, the known and characterized receptors to cryproteins in Lepidopterans are cadherins. Cadherins are the primary receptors to which the activated toxin binds, get oligomerized, and then it binds to the secondary receptors in aminopeptidase as aminopeptidase or alkaline phosphatases. As I had been saying before, I mean, look at the beauty of evolution of proteins of Bacillus thuringiensis. It has selected very key housekeeping proteins as the receptors, so that the organism, the insect, cannot afford to have a mutation. Cannot afford to have a mutation. See, for example, if a mutation is there in aminopeptidase, the BT protein cannot bind, but then it will suffer indigestion. It will suffer, it cannot process its food, because a growing caterpillar, the digestive mechanism has to be extremely powerful, extremely effective. So it cannot afford to have a mutation in any of the enzymes which participates in digestion of food. That's the beauty of evolution of BT organism and its proteins, because it has evolved to multiply within a special niche of with insects below the soil, above the soil. And the BT resistance has also been very nicely worked out. For example, there are many mutants in insects have been seen, where mutations have occurred in ALP or the aminopeptidase or alkaline phosphatase or cadherin. So this is the mode of action of the mode of resistance development is through mutations in any of the receptors. In Bolgard 2 and also in some insects in Bolgard 1, mutations could be developed in the ABCC, that is the ATP binding cassette transporter proteins. These are very important housekeeping transporter proteins, and any mutations in this will render it sort of resistant to the Crivin AC and the Crivin2AB2 proteins. And when we looked at the molecular basis of Crivin AC resistance in the pink bollworm, pink bollworm populations were picked up in Gujarat, in Maharashtra, and in Madhya Pradesh. All these strains were processed, and the mutants were studied. The molecular basis of mechanism of resistance was studied by Fabric et al., and they published it in 2014. What they found was the pink bollworm populations collected from the 3 major states of India. There were 8 novel severely disrupted cadherin alleles. As I said, cadherin mRNA undergoes processing before it becomes a protein, and there were 8 alleles with severely disrupted processing of the mRNA. As a result, 19 transcript isoforms could be isolated, each consisting of premature codon and multiple DNA deletions. So we had issues in the RNA processing of the cadherin. We had issues with multiple deletions in the cadherin gene. As a result, multiple alleles in cadherin could be developed, and they provided resistance to Crivin AC protein in pink bollworm. Now, the sequence of alleles suggests that alternate splicing mechanism resulted in mutated cadherin proteins on the cell membrane. So this was a molecular basis of the resistance to Crivin AC in pink bollworm populations of Rajasthan, of Gujarat, Madhya Pradesh, and Maharashtra. And now, what do we see? I mean, molecular studies have shown that resistance to pink bollworm to crack in Cry2AB2 in 2 strains of pink bollworm was through mutations in the ABC transport subfamily A protein. So that is a new mutations which they have mapped. And surprisingly, in China, what they found was transposon insertion causes cadherin misplicing and confers resistance to Bt cotton in pink bollworm. Transposons are very easy to track, and some transposons leave a copy of it before jumping into the other site on the DNA, which means if it leaves a copy, it is a permanent mutation in the insect. If it exits and moves to some other, it is a transient mutation. So transposon-mediated mutations are also described in the cadherin gene of pink bollworm. And they found this transposon-mediated mutations were a key factor in the resistance in pink bollworm in China. Okay, now we know the molecular mechanism of resistance. Now let's look at what are the options we have before the cotton farmer for the management of pink bollworm. One is the integrated pest management. Yes, this is a very powerful, measures of practices. It can break the pest cycle. For example, the farmer has to involve in destruction of the unopened bolls on stalks, summer plowing to destroy the pupae and diposing larvae, and trapping of emerging adults in gins and destruction of gin waste. I mean, if this is diligently and meticulously done in all the cotton growing areas, yes, we can have a suppression of the resistant strain of pink bollworm. Now, USA has very successfully managed pink bollworm on cotton. How did they do it? One is the sterile male moth release. Now, USDA had began a program several years for the last, I think, 7 to 8 years. And this program involved massive releases of sterile moths, sterile pink bollworm moths every week during the cotton season. And then they also had an effective Bolgar 2 cotton growing there. And they also deployed synthetic pheromones in the form of traps or in the form of mating disruption. And so it was a combination of all these methods, that is sterile male release, effective Bolgar 2 cotton in the field, and synthetic pheromone traps and lures, which really eradicated pink bollworm in India, Texas, Arizona, New Mexico, and California, and which started in 1918. And I think in 2020, or even I think much earlier, they had the USDA had announced that the pink bollworm endemic areas were now free of pink bollworm, and pink bollworm has been eradicated from these cotton growing belt. Now, Oxitec is also experimenting with genetically modified, which is male lethality gene driven pink bollworm, and is being projected as a technology to supplement the dual gene technology cotton. I mean, I'm only trying to show you the technology landscape available for the control of Bt resistance. Now, USDA has announced eradication of pink bollworm following all these methods. Can we do it? Can this be translated to India? There are several challenges. One is the pink bollworm infested area is very large. Now it is almost 12.5 million hectares, containing homozygous resistant population, resistant to both Cry1AC and Cry2AB2. And we have multiple growing cotton growing seasons. For example, in South India, in Telangana and Andhra Pradesh and in Tamil Nadu, we also grow summer cotton, which adds to the population levels of pink bollworm. And we have, the most important thing is we have ineffective BT cotton to pink bollworm. So a major factor, a major impact making factor has been removed. So, following the footsteps of USA for the eradication of pink bollworm is very, very challenging because of these issues. And number two, it is cost intensive. We have to set up gamma irradiating facilities for sterilizing and releasing male moths. And also, this has to be a wide area participation. It is not that if this is done in a few cotton fields, it would be effective. No, it needs a wide area participation, which adds to the cost immensely. Can we do it? Yes, if we have the power, the money power, and the intention, yes, we can do it. Now, what is happening in India currently? Currently, there is a big campaign started by organizations for the mating disruption control of pink bollworm. So sex pheromone applied on cotton plants disrupt the mating cycle by deterring the males from mating with female moths. I mean, the male moths keep going around the cotton plants on which a dollop of the splat has been applied, the pheromone has been applied, and the males get confused, exhausted, and they die. So they really cannot mate with the female moths, which arrive probably a little later. Now, this huge campaigns by the South Asia Biotechnology and Research Organization, and also by ATGC. This is a biotech private company in Hyderabad. They have taken up huge campaigns in the last 2-3 years. Yes, I really wish these campaigns a lot of success. But again, the caveat is it needs wide area participation. It is not enough if just about 50% of the farmers in a village adopt these measures. It has to be on the scale of 80 to 90%. It needs wide area participation. That is really challenging, but these are powerful tools. Let us hope for the best. Now, the question, the conundrum before Hamlet, written by Shakespeare, was to be or not to be. But in our case, to be tea or not to be tea? Should we be involved in producing the next generation BT cotton? That is the question, and that is what we will discuss in the next few slides. In my opinion, in my personal opinion, having experienced BT cotton for the last 20 years, BT cotton is still a strong option for overcoming the present BT resistance and the future management of pink bollworm and all other bollworms, all other insects, probably including sucking insects in the years to come, in the decades to come. If India has to occupy the top spot till 2030, as one of the media headlines said, we have to increase our productivity. We have to increase our productivity, and this is possible only by impactful new technology. Now, I firmly believe BT still is a large reservoir of bioactive molecules, very large reservoir. And we have to mine for pink bollworm effective toxins. Let me tell you, there are 75 families of cryproteins. 952 cry genes have been described, have been characterized. Of course, many of them may not be toxic, but unless we mine for these effective proteins, effective on pink bollworm, on other bollworms too, we will never come to know. So what I'm trying to show you is BT is still a very, very large reservoir of bioactive molecules. We have to start mining and testing these proteins against all these lepidopteran insects. Now, recently described the site proteins. The site proteins, they are membrane lipid binding site proteins. They are very effective against dipterans, lepidopterans, and coleopterans. And certainly we can derive a synergy between the cryproteins, the 952 characterized cryproteins, and the site proteins. The site proteins are also large. And for example, few laboratories have already started engineering Bacillus thuringiensis hybrid proteins, which are active, making use of the the good properties of site as well as the cry. Now,
You have another 5 minutes.
okay, quickly I'll wrap up. In addition to the Bacillus thuringiensis proteins, we have insecticidal proteins from other pesticidal organisms. For example, I have listed about 13 different environmentally discovered organisms. They also produce a lot of insecticidal proteins. So our reservoir for dipping into has enlarged to include from other bacterial species. So can we say now move over Bacillus thuringiensis? I bet not. I think the time has not arrived. Now, the structure of some cryproteins has been very well elucidated. For example, few of the cryproteins have been characterized. Their 3D structures have been very well elucidated. And now we know domain-wise which domain binds to the receptors, which domain makes a pore into the epithelial cell and causes the leakage of contents. The molecular mode of action by domains is well studied in many of the cryproteins, as I show here. The molecular receptors for some cryproteins have been identified in many of these insects, and they have been well characterized. The bioinformatic analysis of these cryproteins have been well made, and it's available for anyone to work with. And now, most importantly, hybrid toxins can be engineered with huge pest control potential. And this can be done through DNA shuffling of cryp genes. Okay, so we know the functional motive of most of these cryp proteins. So we can select the domains of various cryp proteins and make a hybrid protein which could be very active. And some of these companies like Monsanto and Bayer are deep into this technology of making hybrid proteins, and also BASF. So see, there are a couple of methods available and used for providing for generating hybrid crytoxins. One is through domain shuffling, because we know the characterized and functions of each of these domains in each of these cryproteins. So we can resort to domain shuffling. We can resort to rational design of cryproteins. Now, since we know the receptors of most of these, target insects, and we know the mode of action of these domains, can we rationally design cryproteins? It is possible, and this is being done in many laboratories. So in 1970s and 60s, it was a confluence of BT crops, a chance confluence of opportunities, tools and methodologies in genetic engineering, protocols in tissue culture, research in agrobacterium were at the peak. So we had a confluence of many methodologies and technologies available for the development of genetically engineered cotton and other crops like corn and soybean. And many of these technologies are in an advanced stage today. So which means building the next generation genetically engineered cotton will not be a big task, as it was a big task in 1970s and 80s. And yes, after you develop a BT product, BT resistance management is a participatory job of many agencies. And hopefully we have learned from the mistakes. Hopefully we have learned by the experience we have gone through with resistance to Cry1AC and Cry2AB2. And some final thoughts. Now, BT resistance is found in pink bollworm populations of all 8 cotton growing states across 12.5 million hectares. Now, cotton is too valuable a product for India to be lost to pink bollworm. Let me repeat. Cotton is too valuable a product for India to be lost to either pink bollworm or other bollworms. The worrying factor is that pink bollworm is appearing early on bloom even at 50 days today. Integrated pest management methods, including mating disruptions, are powerful tools to disrupt the pink bollworm pest cycle, but have to be adopted diligently on a wide area scale. Is this possible? Now, this is a big challenge considering the large clientele of 7.5 million farmers growing BT cotton today. Now, most importantly, considering the currently available biotech approaches, the scientific capability and capacity in India, developing a new BT cotton with stacked BT proteins. I mean, individually, these BT proteins have to be effective on the current resistant pink bollworm strains, is the easiest and fastest way to address the pink bollworm issue for the future. This is the only way I see to address the pink bollworm issue for the future. Now, the caveat is, I hope we have learned lessons in the better resistance management and strict trade quality stewardship of BT cotton seeds. Now, the discovery and product development pipeline for BT cotton is well standardized today, not what it was, say, 20 years back, 15 years back. It is well standardized. And now we can predict the timelines of each of these phases, from discovery to regulatory. Now, which means my take-home message is, it is imperative that Indian laboratories get back to the drawing board, probably on a mission mode, probably on a consortium-based approach, to develop new set of BT cotton transgenics effective on key cotton bollworms and sap-sucking insects. Now, I have kind of mapped the various phases of development, right from discovery to phase 4, and each phase involving a lot of activities, trans-laboratory activities. And what I have kind of calculated is, it would take 5 to 8 years to market if we develop today, and it would be around 400 crores for one BT cotton product, which could be effective to pink bollworms and other bollworms too. Now, if we have to kind of include all other technology packages in this, it could be a little cheaper than, say, 800 crores. Thank you very much for your time. Thank you very much. I'm not sure if I overshot my time.
Thank you. No, it's okay, Mohan. I think you did a wonderful exposition, a detailed one, tracing in a sequence how it developed. I think thank you very much, and I'm sure the participants would have enjoyed listening to it. Now, I think we have about 15 minutes for interaction or discussion. I think at the end, you actually left with the feeling that the glass is half full, because the technological options of sterile male techniques or gene drive, pheromone-based mating disruption, new BTs, either discovered from native populations of bacillus or modified using the new techniques that we have today, modified cry, for example. I think all hold promise. So I already have several questions in the chat box. And can everybody put their microphones on mute? Thank you. Let me start with a question from Narendra Singh. This was the first question. You talked about gene drives, sterile male techniques. You talked about new cry proteins. What is the opportunity for gene editing technologies, say CRISPR-Cas, in developing some of these new tools?
Yeah, gene editing, there are no leads at the moment. If gene editing could be used for for breaking the BT resistance in the bollworms, there are no such leads. I've been going through the literature. And also, gene editing is more effective for gene knockouts. Now, if we can identify and knock out gene, which acts as a suppressor for a few secondary compounds, which can provide resistance to these bollworms, yes, it is possible. I'm only trying to imagine a scenario, but there are no leads at the moment?
Yeah, I suspect in modifying the existing cry genes to bypass the non-binding in the receptor, or in designing new cry genes. Of course, knockout is one system, but now you have the prime editing and base editing. So maybe as these tools are refined, they may find an application. Let me move on to the next question. This is from, I could only read this one. It said agribiosis. I don't know the name of the person, but he has asked whether what is the role of phenolics in allowing for resistance development in the pink bollworm and other bollworms. Is the low level of phenolics responsible for I think he is suggesting chemicals or compounds like gossipol. Are they responsible for the fast development of resistance? Any evidence or any relationship?
No, I would say on the contrary, the bollworms have evolved over tens of thousands of years to overcome all these plant phenolics and secondary metabolites in the cotton plant. As you know, cotton plant produces a lot of phenolics. Okay, so by enhancing the level of phenolics, I think it's a very difficult path for gaining resistance to bollworms.
Let me go to a question by Chetan Sinde, which is again posted. He is wondering whether you can suppress the resistance development in these bollworms or other insects by epigenetic modifications. Don't modify the sequence as such, but I know modify their expressions both spatially, temporally, through methylation or means like that.
You mean these epigenetic changes in the insect?
Epigenetic changes in the plant, I guess.
Yeah, yeah.
Is Chetan Shinde in the audience now? Chetan Shinde, you had posted a question in the chat box.
Yes, sir.
Yeah,
Chetan, you want to ask that question directly to Mohan?
Yes, sir. Can it possible to make a epigenetical modifications at plant level as well? To revert to or to...
Okay, so Mohan, he wants you to tell you whether plant level the genome can be modified epigenetically to address the issue of resistance development.
Yeah, I need to let my imagination run wild.
Yeah, you can do that.
Okay. Yes, it is possible. It's possible. And provided we find out the targets. If we find out the target genes, if by methylation or, other methods, you induce changes in the DNA molecule, if those target sequences can affect resistance development, yes. But first of all, we need to find out those target genes. Yes, it is a possibility, a very distinct possibility. And as I said,
let me add on in thinking a little more once you said that. In the plant, if you generate epigenetic changes, it will change the expression of the Bt protein which the plant expresses. It will either suppress the expression, it may not change any base pairs or anything to modify the binding properties. Whereas in the insect, if you change it, it may change the expression levels of some of the receptors. Maybe you have to think more deeply about it. And I think, like you said, and then let the imagination run.
Thank you.
Okay, I think now let me just switch gears. I think we have experts like Dr. Manjunath and other people in the audience. I think he has spent a lifetime, as many of you know, in the biological control of crop pests. Dr. Manjunath, are you there?
Yes, he's there. He's there, Narayan. I can see him.
Yes, I'm here.
Okay,
Yeah.
Okay, sir. I think the question is, what I have is, the pink bollworm is the immediate problem. Helicoverpa could be developing resistance, but so far it is manageable.
Yeah.
Are there any predators, parasites that you have or you know of for pink bollworm, Pycnophora gossypilla, and what is the prospects if there are none as of now? What is the prospects for discovering them in future, and what we should do for that?
Mm, okay. Can we move on from the entire screen can be seen?
Yeah, can you take off your presentation, Mohan?
Yeah.
Okay.
If you can stop sharing, I think he can start sharing.
Yeah, stop sharing so that we will see the people on the screen. You have to stop sharing.
Yeah, is that okay?
Yeah.
No,
that's
you are
okay.
still
Stop.
sharing.
Stop sharing.
Still, we are
Stop.
seeing your screen more. Yeah.
Yeah.
No,
Okay, that's
now we
better.
can. Yeah.
Yes, Dr. Manjunath, you can now. Yeah, you are now in the center of the screen.
Okay. With regard to your question regarding biological control, of course, a lot of natural enemies have been discovered and also produced and used. But in a cotton situation, for example, trichogramma has been recommended for controlling the bollworms. It's a common parasite for all the bollworms. But although it has been recommended in a cotton crop where there are multiple pests, it would be very difficult to refrain from spraying insecticides. At least for the present, we have to use insecticides for controlling the sucking pests. So when that is used, the natural enemies will be destroyed. Even if you make releases, it will be destroyed. So I don't see any scope for biological control at this stage, although a large number of natural enemies have been discovered. There are parasites, predators, and also pathogens. So this is my answer to it. And then with regard to Dr. Mohan's final recommendation, I would like to say that the sterile male technique involves lot of massive production of the moths, pink bollworm moths, which is very laborious, and also it is difficult to make that kind of a release. So in my view, mating disruption is a very viable option because here the pheromones can be produced in a factory and it can be used, and it has also been demonstrated in the USA, in Greece, and a number of other countries. So with regard to its using, as Dr. Mohan mentioned, that it has to be used on a large scale, I think if we depend on farmers, it will not become a reality. The government has to take this program as a farmer welfare program and try it out on a large scale, at least for 2 to 3 years, so that the bollworms can be brought down. And if the government is not willing, when I say government, it can be central government or it can be state governments. And if they are not willing, then probably the Cotton Growers Association can take it up as a challenge and then do the entire process in the interest of the farmers. So this is my opinion. And then and another option is with regard to using refuge. Dr. Mohan, probably we are trying to use refugia in order to allow the susceptible moths to develop. Probably one area where we can give thought to is that we can actually use the pheromone lures which attack the males. And those males, the recommendation is that they should be trapped and destroyed. So instead of destroying them, those males, they come from distant places. Instead of capturing and destroying them, I think they should be allowed to escape so that they become a source of susceptible moths. And this actually, we can avoid dependence on refuge crop. I don't know. I would like to hear Dr. Mohan's view on this. Trapping them, instead of trapping them, attracting them and allowing them to escape and mate with these, mate with the males which have developed on BT cotton. So these are some of the thoughts that I have in my mind.
Thank you, sir.
So-
I think, Mohan, now you have to respond to some of those issues. Before I go to people who have raised their hands.
Yeah, quickly I'll respond to Dr. Manjunath's comment. See, currently, even if you trap the males, I believe the homozygous resistant population is so high in the environment today, whatever you trap again will be all BT resistant males.
Okay, that's a quick answer. I'm sure we cannot resolve many of these issues in this short discussion. We can have maybe ATPBR should think about having another round table on addressing these issues in collaboration with some of the South Asian Biotechnology Association and all that, who are doing human work in the field. Dr. Krishna Kumar, you have raised a hand. Probably you have a question or a comment. No, you are on mute, sir.
Dr. Mohan, thanks for a very nice talk and very informative talk. Let me quickly come to some two quick points. We are looking too deep into molecular and too shallow ecologically. The reason that I'm trying to tell you is ecologically there is a need to understand the population genetics of the resistant populations vis-a-vis their dilution, amplification, migration, random mating. If we have to really address, and some school must address at least with regard to pink bollworm and some of these things that we are talking, number one. The second one is, you made a general statement regarding sucking pests at the end of the day. Which sucking pests are we talking? Which, according to you, should be the prioritized? What are the trade-offs that you think that is parallelly that's going to come? Lastly, the third small question is, should a variety be released only after we are reasonably robust with regard to its resistance management?
Oh, yes. For example, I'll probably answer your last question first. I mean, before a BT product is launched, okay, the pre-launch stewardship studies involves the calculation of the risk of resistance development. See, resistance development in the target bollworms is contributed by many factors, including the genetics of the insect, including the high dose expression in the cotton plant, then the feeding behavior, mating behavior, so many factors. So yes, an assessment of the risks have to be done prior to the release. And I think you
ecology also, versus molecular approaches.
And yeah, I think the second question ecology was which are the sucking pests of cotton you have in mind while developing a technology. I think many of these insecticidal proteins, even from BT, have been known to have action against the hemipteran insect. For example, BT cotton plants have been developed against ligus, which is a hemipteran bug, Ligus hesperus and Linolaris. So it is possible, and there are reports of toxicity of some of these not so common BT toxins to these hemipteran insects. Yes, it is possible. And also probably we should bundle up a technology to break the symbiosis between the whitefly and the virus. That is possible through gene editing technology or RNAi technology. Yes, these are things we need to think upon, develop, and bundle it into a single plant. And on the ecology, yes, ecology is extremely important. Probably we had, not paid enough attention to ecology whenever we release a new BT. This thing, we really do not study in depth the kind of imbalance it would produce in the ecology of predators, parasites, and the pest.
Thanks, Mohan. I think we are running almost short of time, the tyranny of the clock. But I saw Rajiv had raised his hand long back, and I will give the last question to him before I wind up with some final remarks. Rajiv, you want to go ahead? Rajiv Tagar?
Yes. Thanks, KK. Thanks, firstly for organizing this, and I have got 5 or 6, points. I won't take time, but can I be quickly enumerate, this?
If you can make it within 30 seconds, like they say in the TV debates.
Okay, right. The first thing is, I think, do
you prioritize your 5 questions? Ask the first question first.
No. These are points rather than questions, I would say.
Okay,
Dr.
go
Mohan,
ahead.
thanks for everything. Yeah, thanks for everything. But the thing is, I find this presentation highly one-sided. There are a lot of points, pardon me for saying this, there are a lot of points I would like to mention here. First of all, I mean, the whole presentation is based on vertical resistances. We are forgetting totally that pink bollworm, in your own words, came into being as early as 1918. I have seen theses written, PhD theses written in 1964, working on pink bollworm resistances in cotton plants in both in USA and even in India. In IRI, there's one from 1970, a PhD thesis. Now, coming back to, because we all know about boom and bust cycles, these resistances in plants have been building and going down, building. So resistances are definitely there. We are not even looking at the surface of the germplasm, the resistances which is naturally existing. No, but the important thing is, I mean, we need to look at both. This is point one, vertical and horizontal resistances. So far, we are talking only about monogenic vertical resistances. There's a very good point which came from agribiosis, which was not really point very well taken. I'm sorry to say. The person talked about the phenolics. It's not just phenolics. We are talking about a lot of hormone-mediated mechanisms of horizontal resistance. We are talking about jasmonic acid. We are talking about strigophenols. We are talking about, let's say, there are other lot of, so all those mechanisms of horizontal resistance, they are totally missing in our present scheme of things. For the past 2 decades, we have thrown it out of the window, not at all touched it on. Okay, the other thing is, I mean, we are reinventing the wheel. I mean, this boom and bust cycles have been, so that is just my one point, that we have totally ignored the horizontal resistances. We are stuck only to vertical resistances, and the whole pessimistic viewpoint that only BT can take over it. I think, like I said already, I find it extremely pessimistic. The plant itself has been doing it many times over and over in the natural history, starting from 1918.
Thanks, Rajiv.
Okay.
I think that's a perspective, and we respect it.
Okay.
Mohan, I will save you from answering that. I will just, because I will go into making my comment. That were useful comments. Whether there is a natural resistance in the cotton population, horizontal resistance for pink bollworm that needs to be explored, whether it has been discounted, whether people have discarded it for going into more sexy kind of approaches like BT. I don't know. We can debate on that endlessly. But I would like to leave that on the table for people to decide. And what I would like to do is now to wind up. We are already 12:28. What the clock is showing.
Dr. KK, can I get a 10 seconds time? I am Dr. Vajita, Director CACR Nagpur.
Okay, 10 seconds time, sir. Certainly, yes.
Dr. Mohan, very nice presentation as usual, and very nice to hear that you still believe in BT, and that we can still come out with something BT, some innovative toxin for pink bollworm as well. I just wanted to ask you a brief comment on stewardship. How do we really take forward this BT? You still believe in BT? Second is, there is a lot of talk that COT102 Syngenta gene can still be effective, and it is being marketed in Gujarat as pink BT or something like that. How effective it is. Third is about, I heard that there is a non-BT protein application.
It's now already 40
US,
seconds.
which is already filed.
Quickly.
And the fourth one is the drawing board coming back, stakeholder consultation, and because a lot of investment is needed. I would like you to facilitate that, because you have been in research as well as in industry. Thank you.
So I think
thank you, Dr. Prasad. Thank you very much.
Give a quick answer, and then rest of it you can have a direct communication with Dr. Enumula.
Yeah.
Okay.
Sure. Dr. Prasad, the first on the stewardship, yes, we have learned a lot of lessons in the stewardship path of BG1 and BG2. I am sure we can make it much more robust. And number two is WIP-COT, which has WIP3A, is only moderately toxic to pink bollworm. It's only moderately. And now the current strain of resistant pink bollworm will have to just face only one additional moderately active protein. It will quickly gain resistance, even if that particular event is brought into India. No, that is not going to be a solution. Within a couple of years, they will gain resistance. And that too, we have not field tested to see if that particular event will be active on the currently present strain of pink bollworm. We don't know.
Okay, thanks, Dr. Mohan, and thanks, Dr. Prasad and others who interacted. Bala and others who have raised their hand, I'm really sorry, we are running out of time. So I think you will all join presentation on how the BT cotton became susceptible to the pink bollworm, the mechanisms of resistance development in the insect, and some of the light at the end of the tunnel on terms of what additional methods that we can employ to address this problem in the future. I will only end by saying this. This is a huge problem, and so we need to have multiple approaches, a multi-pronged approach. It is not that we are trying to promote one approach over the other, but BT continues to be a very potent tool in the armory that we have. We need to look at host plant resistance, like Rajiv was suggesting. We need to look at, mating disruption, sterile male techniques, modified cry-C. All these have to be looked at. For a vast country like ours, where more than 12 million hectares of cotton is grown, and we know that every year, if you look at the total cotton economy, the additional value that is being created by the resistance to the bollworm using BT technology has been estimated to be about 40,000 crores, which is going to all the 7.5 million cotton farmers or somebody. What the seed companies harvest out of that is a small proportion, which is about 4,000 crores. So I think we need to keep this going, and I will try to reiterate what Mohan said, that we cannot dump any technologies, including BT technology. It has a future. We need to be creative in finding solutions for the future. Thank you very much. I now leave it to Aparna to wind up this session.
Thank you, sir. Thank you very much. Thank you, Dr. Mohan, for such a wonderful presentation and a great discussion afterward. Unfortunately, there are a lot of questions are still in the chat box. Maybe, I'll pass them, and in your spare time, you can check and revert to those questions. And thank you all the participants for all your time, and looking forward to see you again next month with another webinar. Thank you very much. Have a great day.
Thank you very much. Thank you.
Thank you. Thanks to the host organization.
Thank you.